Okay, in this video we are going to finish up our chapter on the cell. And so last time we finished by talking about passive transport. So now we're going to talk about active transport. So if you remember, with active transport, it is going to require ATP. Okay, so it requires ATP, and typically we're going to be moving things against their concentration gradient, right? So from low concentration to high concentration, and that's a process that requires energy. Okay, and so one of the major examples of active transport that you should be familiar with, because we're going to use it over and over again, is going to be the sodium-potassium pump. Okay, and so we talked about before how there's different concentrations of sodium on the inside of the cell versus on the outside of the cell. So sodium is generally going to be high, outside of the cell. All right. And potassium is going to be high inside of the cell. All right, relative to the outside. So sodium is higher on the outside than it is on the inside, and potassium is higher on the inside than it is on the outside. Okay, and so the reason for this difference in concentration is because of this sodium-potassium pump. Okay, and the sodium-potassium pump, its job is to take sodium, all right, and it pumps sodium out of the cell. And then as it's doing that, at the same time, right, it's going to grab potassium from the outside of the cell and move it inside of the cell. Okay, so this is created, right, that difference in concentration, it's created by the sodium-potassium pump. Okay, so this is just a membrane protein, and what it does is it will, if you think of it kind of, as like a little mouth, that mouth will open up to the outside of the cell. It'll grab sodium, all right, and then that mouth will flip the other way, open up to the inside of the cell, release those sodium into the cell, pick up potassium, and then it'll flip again. It'll open this way, it'll let go of those potassium, pick up more sodium, and it'll just keep doing that over and over and over again. Every time it does, it's going to require ATP, right? Every single exchange like that requires ATP. Okay, so we'll be using ATP here every time we move some potassium in and sodium out. Okay, and so specifically you guys should know how many of each are moved with each pump of that sodium-potassium pump, right? So we're going to move 3 sodium out every time, and we're going to move 2 potassium in with every pump of that. All right, and each one would require, each pump would require ATP. Okay, and so then they're moving against the concentration gradient because we have a lot of sodium on the outside. We're taking sodium and pushing more outside where there's already a lot. And we're taking potassium and pushing it to the inside of the cell where there's already a lot. And so that's why it requires ATP. Okay, and so this difference in ion concentration that's established by the sodium-potassium pump is going to be essential to many processes in the body, including muscle contraction, nerve function, nutrient absorption in the digestive tract. There's going to be many things that you're going to see are going to rely on this concentration difference of sodium and potassium. Okay, so sodium-potassium pump is extremely important. All right, so that is obviously active transport. It requires ATP. And then we have a couple of kind of less obvious examples of active transport, because you can't really see how, we're moving against the concentration gradient or anything like that. All right, so just think of, so this is a category called vesicular transport. All right, so we're going to be taking vesicles, right? So, those transport vesicles or secretory vesicles in the cell, and we're going to be moving them. So we're either going to have a vesicle, we're going to move it out of the cell, or we're going to be taking something in, as is happening here, right? So the cell's eating something and moving it into the cell. Both of those processes require energy, and that's because you're moving these large quantities of material into or out of the cell. And to move that large of an amount of material requires energy, okay? So it's not as much based on the concentration gradient, but more so the amount of material that is being moved. Sometimes this is also called bulk transport, okay? Because you're moving large amounts of material. Okay, so let's talk about the two types. So we have endocytosis, right? This would be taking things in. Okay, so eating, or we're going to see drinking things, right? So we'll say taking material in. All right, and then exocytosis would be secreting something. So we're going to be releasing material, right? Release or secrete material. So this would be where our secretory vesicles would come into play. So let's talk about endocytosis first. All right, there's two types of endocytosis. So first is phagocytosis, which we're seeing here. And so phagocytosis is just cell eating. Okay, so you have cells in your body that are called phagocytes or macrophages, and their job is to just go around eating debris, or in this case, this would be like a little bacteria, so a pathogen, and digesting them and getting rid of them. Okay, and so what it's going to do is it will take part of its cell membrane, and it'll start to extend it around whatever it's trying to eat, right? Until it extends it all the way around and completely encircles it with cell membrane. All right, and then this little kind of packet of food, right, bacteria, debris, whatever it is, will be moved into the cell, all right? And then it will be merged with a lysosome, which will help to digest the contents. Okay, so phagocytosis is cell eating. There's also another type of endocytosis called penocytosis. And penocytosis is cell drinking. All right, so I always remember that just because, like Pinot, the wine, right? You drink wine, so penocytosis is the cell drinking. And so for this, instead of taking up large either cells or debris, pieces of debris, the cell is just going to sample the extracellular fluid. It's just going to be drinking some of that extracellular fluid. Okay? So there are two types of endocytosis. And then exocytosis, we briefly talked about this with our secretory vesicles, is going to be our secreting of materials. So the cell will make different things, maybe a protein or a hormone, or if we're talking about a nerve cell, this would be like a neurotransmitter used to communicate between nerve cells. And it's going to take that vesicle, that secretory vesicle, and move it close to the cell membrane until it merges with the cell membrane and its contents are released. All right, so secretory vesicle is sent to the cell membrane. Okay. All right, and so remember, all of these processes, endocytosis and exocytosis, do require ATP. Moving big vesicles around the cell like that requires ATP, so that's active transport. All right, so our last topic for this chapter is on the cell cycle. Okay, and so the cell cycle encompasses basically the entire life of the cell. Okay, so when the cell is first kind of quote unquote made as it grows up and it, performs whatever its normal function is, and then it divides into new cells. All right, so that whole process is going to be the cell cycle. All right, so we divide the cell cycle into interphase and mitosis. Okay, so interphase is kind of everything leading up to cell division. So this is everything prior to cell division. Okay, and then mitosis is the actual process of cell division. So we're going to take one cell and divide it into two new cells. Okay, and so the whole point of mitosis, this cell division, right, is that we want to make new cells that are identical to the original. All right, so identical to the original. Okay. All right, so let's talk about interphase first because it's a little simpler. You don't have to know quite as much of what's going on, and then we'll dive into cell division and then the steps of cell division and what's going on with that. All right, so during interphase, there are three kind of separate phases within interphase. Okay, so we have G1, which is the first part. And so G1 phase is just where the cell is growing. So this would occur right after the cell has previously divided. So that cell needs to grow, make more of, the important proteins and organelles and stuff, and, get up to its normal functioning size. Okay? And then kind of later in this phase, so it would be growth, and we'll just say growth and normal function. Because later in G1, the cell is grown and it can carry out its normal function. Okay. Interestingly, there are some cells, not many in the body, but there are some cells that don't divide, right? So once you have that cell and it's mature, it does not divide anymore. And so those cells would stay stuck in G1 phase. They wouldn't move past this phase. And so some cells that don't undergo mitosis, right, they're stuck in G1. Those would be neurons, right? So neurons never divide. Once you have a neuron, that's it. You don't get a replacement neuron, right? And actually heart cells typically don't either, right? That's why when heart muscle dies, it's so detrimental because it's not replaced. You don't replace heart tissue. They don't divide and make new heart cells. Okay, so those types of cells would be stuck in G1. All right. However, if a cell is going to divide, it will then move to the S phase. In the S phase, think of S as meaning synthesis, right? So we're making things, because here we are going to duplicate our DNA, right? All of the chromosomes. We're going to duplicate the DNA, and we're going to duplicate the centrioles. And so if you remember from earlier in this chapter, centrioles, their function is to make microtubules. Specifically, they're going to make the mitotic spindles that we're going to be seeing in a moment when we talk about mitosis. So normally a cell will have one pair of centrioles, right? But for mitosis, we need two pairs of centrioles. So they're going to duplicate. So we now have two pairs. All right. And then the last part of interphase is G2 phase. And so here we're going to make a bunch of enzymes that are needed for mitosis. All right. So enzymes for mitosis, as well as any other final preparations. So duplicating any additional organelles or structures that you need for mitosis to occur. All right, so final preparations before the cell is going to divide. All right. Okay, so now let's talk about mitosis itself. Before we do, let's go over the structure of a chromosome. So when we talked about DNA before, when we were going over the nucleus, we talked about how when the cell is not dividing, right, the DNA is in the form of chromatin. So it's loose, right? It's not tightly packed. And then when the cell is going to divide, that DNA will condense into a chromosome. And so it does that so that you can easily separate the DNA into the two new cells without this big mess of chromatin that's all kind of, wrapped around each other and tangled up, right? So it makes it really easy to separate them into two different cells. Because each new cell is going to need an identical copy of the DNA so that you'll end up with absolutely identical new cells. Okay, so our chromosome, the structure of it, is that in the middle, all right, we have this centromere. Okay, and that centromere, its function is that it holds together both of these duplicated sister chromatids. All right, so that's one of our new words, right, as a chromatid. Okay, so each of these would be a chromatid, okay, and so they're absolutely identical, okay, so they're duplicates of each other, the chromatids. Okay, so each one of these is an exact copy of a specific part of the DNA. Okay, and so they're held together in the middle by the centromere. So that centromere is holding them together. All right, so it holds the chromatids together. Okay. All right, so that's our chromosome. So let's go through the stages of mitosis. All right, so you should know these stages and generally what is going on in each stage. All right, so the very first stage or phase of mitosis is called prophase. All right, so when we go from, so this would just be representing interphase, right? And then we're transitioning to prophase, which would be this cell here. Okay, so when we move from interphase into prophase, there's a few events that happen. I'll write them off to the side here. All right, so the first thing that's going to happen is that the centrioles, all right, remember we duplicated them, the centrioles move to opposite sides of the cell. All right, so we're going to move to opposite sides. Okay, and so you can see here, right, this is one set of centrioles. Yellow is not super visible, right, but here's one set of centrioles. Here's another set, right? At some point they've duplicated. Maybe they're sitting here together, duplicated, and then one set moved here, the other set moved here. So they need to be at opposite ends of the cell, okay? So they're going to move to opposite ends of the cell, and they're going to start making those mitotic spindles, or spindle fibers, you may see them called. Okay, those mean the same thing. All right, so start making mitotic spindles. Okay, and so again, those mitotic spindles, all right, would be right here. So there are these green threads coming out of those, remember they're made of microtubules, out of those centrioles. All right, so this one's gonna be making some, this one's gonna be making some. Okay, so that's happening. All right, also the chromatin, all right, that was in the nucleus, chromatin, remember it duplicated in our S phase. Okay, so we've duplicated all of our DNA, all of our chromatin. Now we're going to condense that chromatin into our chromosomes. Okay, so it's going to condense to the chromosomes. So then each duplicated piece is going to become one of these duplicated chromatids. They're going to be held together in the middle by our centromeres. Okay, so that is going to be here, right? We have our little chromosomes here, you can see. So our loose kind of DNA chromatin spaghetti here is now condensed into these little chromosomes, a bunch of little X's. All right, and then lastly in prophase, the nuclear membrane is going to start to dissolve. All right, nuclear membrane or nuclear envelope is going to start to break apart. Okay, and the whole reason for that, all right, let's try a color you can actually see. There we go. Okay, so the reason for that is because, remember, we're going to take and divide this DNA. If the nuclear membrane is there, we can't divide the DNA. It's going to be in the way of that process. So we need to get rid of that nuclear membrane so that DNA can be pulled apart to the two new cells. Okay, so that's prophase. So the three kind of steps or things that are going on, events that are happening in prophase. All right, so next we have metaphase. So this is our second kind of phase of mitosis. So, all right, kind of the events of metaphase here off to the side. And so with metaphase, it's pretty simple, right? There's not anything complicated going on. And I always remember metaphase starts with an M, right? And what's happening in metaphase is that the chromosomes are lining up in the middle. So M, metaphase, think middle. Okay, so the chromosomes line up in the middle of the cell. Okay, so they're going to line up in the middle of the cell, and you can see that here, right? They're all lined up kind of on the equator of the cell, right? So in the middle of the cell. It actually has a name. It's called the metaphase plate. So they line up along the metaphase plate, and you can see that the spindle fibers are attaching to those chromosomes. All right, so the spindle fibers attach to the centromere of the chromosome. Okay, so they're going to attach to that middle part that's anchoring the chromatids together. All right, and actually this attachment of the spindle fibers is what's responsible for helping these guys align on the middle of the cell, right? They don't just magically float to the middle of the cell. These spindle fibers attach, and they'll push, and this one maybe will pull a little, or vice versa, until those chromosomes kind of align in the middle of the cell. Because remember, microtubules, their function is movement. And so in this case, we're moving those chromosomes around. All right, so that's metaphase. Very simple. We're just lining things up in the middle of the cell. Okay, and so then in anaphase, that's our phase 3 is anaphase. What's going to happen is we are going to separate our sister chromatids. Okay, so those duplicated sister chromatids are separated from each other. Right? Remember, they're identical copies of DNA, and we want each new cell to get one of those identical copies. Okay, so we're going to pull them apart towards opposite ends of the cell. For that to happen, that centromere in the middle has to dissolve. So that happens in anaphase 2 also. That happens in anaphase also. There's not a part 2 of anaphase. Right? So that centromere dissolves, and the spindle fibers pull on those chromatids to pull them to opposite sides of the cell. Okay? So they're going to be pulled by the spindle fibers. Okay, so that is anaphase, right? They're going to be pulled to opposite ends of the cell. Oh, I was trying to figure out what that noise was. Sorry. My husband's outside doing yard work. Okay. All right. So that kind of startled me. I don't know if that sounded weird on the microphone. Okay. So that's anaphase. All right. And then we move into our final phase, which is telophase. Okay. And so with telophase kind of overlapping with telophase, and so this might get a little confusing because a lot of textbooks show this very differently. Right, so if we had, I'm just going to illustrate this and then I'm going to erase it, right, so it doesn't get in our way, right? So if we drew each phase that's happening kind of one after another, right, so this is prophase, and then we move on to metaphase, lining up the cells in the middle, and we have anaphase, we're separating our chromatids, and then we have telophase. All right, telophase is actually going to kind of overlap with this process called cytokinesis. Okay, so there's like this overlapping time where they're both happening at the same time. And so a lot of books will describe these as two different events. So you always have cytokinesis happening right after telophase. But when you look at the cell that's dividing, when it's in telophase, obviously cytokinesis is happening. Okay, so that might get confusing. Some books make it part of telophase, right, cytokinesis. Some books make it completely separate, but actually they're kind of overlapping and happening at the same time. Okay, you don't have to worry that much about that. You just have to understand what's happening, for each of them. Okay. All right, so I'm going to move, hold on, cytokinesis down here really quick so we can write a little bit about telophase first. Okay, so with telophase, all right, we're going to kind of start to prepare to divide the cytoplasm of the cell. So we've divided the DNA of the cell, all right, and so we're going to kind of get final preparations to divide the cytoplasm, okay? So the DNA is now on opposite sides of the cell. It is going to kind of uncondense, right? So the DNA uncondenses, right? It starts to go back to that kind of chromatin form, right? And then the nuclear membrane begins to reform around that DNA. Okay, and then also occurring during, remember I just said overlapping kind of with telophase, is this cytokinesis. And so cytokinesis just means that you are dividing the cytoplasm. Okay, and so the way this actually works, and y'all don't have to know the names of these structures, but I think it kind of illustrates it well, is that this kind of protein ring forms around the cell, right? So if this cell were not, divided like this yet, the ring would form like this. And I like to think of it kind of like a rubber band that you slowly tighten. And so that ring it's called a contractile ring. So it contracts and it gets smaller and smaller, slowly pinching between the cells until it's completely pinched all the way through the cell and divided that cytoplasm completely in two. Okay, so cytokinesis, we're dividing the cytoplasm. All right. Okay. And so then we end up with two identical cells. Okay, so producing two identical cells, right? Because they have identical copies of DNA, right? They each have a pair of centrioles, right? We've divided up all of our organelles, our cytoplasm. And so now these cells can start to grow. So they'll go back into G1 phase and they'll grow and they'll start, being able to perform their normal functions. Okay? All right, so those are the stages of mitosis. And so let's just briefly talk about cancer because it is very tightly linked to mitosis. And so it's really important to kind of understand mitosis to be able to understand what is going on with cancer. Okay, and so normally cells have this kind of innate ability to stop growing when they touch each other. Okay, and that's called contact inhibition. Right, so growth and mitosis in normal healthy cells, right, stops when cells touch. Okay, so think about like your skin, right? You have lots of, cells making up your skin. And so those cells, when they're growing, when they start to touch each other, they'll stop growing. And that way you don't make too many cells and it doesn't get too crowded. Okay, and so that's normal for cells to have. With cancer, right, this kind of contact inhibition is lost, right? So they're lost with cancerous cells. Okay, so those cancer cells lose that contact inhibition. So even though they touch each other, they're going to just keep undergoing mitosis and dividing and making more cells. And that's how tumors form, right? These large masses of cells, because those cells are essentially growing out of control, whether they're touching each other or not, they just keep on dividing and undergoing mitosis. Okay, so cancer by definition is uncontrolled mitosis. Okay, and so obviously there's lots of different types of cancers, so many different types of cells can become cancerous. However, one kind of caveat for that is that those cells have to themselves be able to undergo mitosis. So for example, it is not possible to have cancer of a neuron, right? So neurons don't divide, they don't undergo mitosis, so you're not going to have a cancerous neuron. That being said, there are many types of brain cancer, and that's because the cells that support neurons, they're called glial cells, right? We're going to learn all about them at the end of the semester. They do undergo mitosis, and so you can get cancer of those cells, right? But those are not the actual neurons in your brain. And that's why, like, one name, for a brain cancer is a glioma, right? Because that is the glial cell that is growing out of control. Okay, so cells have to actually be undergoing mitosis in order to reach this cancerous state, right? To be able to go into this cancerous state. Okay, and so you know that cancers can be benign or they can be malignant. All right, so a benign tumor is a tumor that's growing, but it's not spreading. Okay, so the tumor, it grows, but it doesn't spread. Whoops. All right, doesn't spread. And by spread, I mean spread to other locations, other tissues. All right, so it stays wherever it is growing. Malignant, on the other hand, is going to be a tumor that can spread to new locations. All right, so it spreads to new locations. All right, or it can spread to new locations. Okay, and so we use the term metastasized to kind of describe this. So you say a tumor has metastasized when that tumor has appeared in a new location. All right, so you found cancerous cells in a new location where it originally was not. Okay, so that's kind of just a very general overview of cancerous cells. All right, and obviously cancer is very complicated, and cancer cells, there's a lot of complicated stuff known about them. It depends on the cell type, and there's also a lot that's still not known about them yet. All right. Okay, and so our last topic is just a very brief overview of meiosis. So you're going to go deep into meiosis in A&P2 when you do reproduction, right? We're just going to kind of compare and contrast it with mitosis. We're not going to go into detail with the steps or anything like that, okay? So the purpose of meiosis is to produce sex cells. Okay, and so by producing sex cells, I mean producing the sperm and egg, or egg, right? Because obviously, right, they wouldn't be produced in the same individual. Okay, so you're going to either produce sperm or egg depending on the sex that we're talking about. Okay, so that's the purpose of meiosis. All right, and so with cells, we say that they're either haploid or diploid. So a haploid cell has only one set of chromosomes. All right, and diploid cells have two sets of chromosomes. Okay, and so most body cells are diploid. Okay, so let's talk about that for a second. All right, so the two sets of chromosomes, they're coming from mom and dad. Okay, so you get one full set of chromosomes from mom, and then you get a full set of chromosomes from dad. Okay, so every single one of your body cells is going to have both those sets of DNA, a set from mom and a set from dad. Okay, so you have two sets, one from each parent. All right, so we usually will kind of abbreviate this as 2N. So if you ever see 2N, that just means we're looking at a haploid individual. All right. Okay, and so that's a total of, in humans, right, 46 chromosomes. Okay, and so that means that you get 23 chromosomes from mom and you get 23 chromosomes from dad. All right, so diploid cells would have that. And so we call these homologous chromosomes. Whoops. All right, so these are chromosomes from mom and dad that code for the same thing, right? So that contain the same type of information. Okay, so from mom, just as an example, right? From mom, you would maybe, if your mom had blonde hair, she would pass on to you the gene for blonde hair on a chromosome. Your dad maybe has brown hair, he would pass on the gene for brown hair to you. So you would have one blonde hair chromosome, you'd have one brown hair chromosome, but it's the same type of information, right? So it's all hair color, okay? It's just slightly different depending on, which parent you got it from. So those are homologous. They have the same type of information. You just get one from mom and one from dad. Okay, so then haploid cells only have one set of chromosomes. And so these are going to be the sperm and the egg. All right, so the only cells in your body that are gonna be haploid are gonna be, either the sperm or the egg. And that's because when fertilization occurs, right, you unite sperm and egg, you want to end up with these diploid cells. So you need to start with half the amount so that when you combine them, you end up with haploid or diploid, right? So this is 23 chromosomes in a haploid cell. So when you combine an egg with 23 chromosomes with a sperm with 23 chromosomes, you end up with a diploid cell that has 46 chromosomes. Okay? All right. So with mitosis, all right, just to give this kind of an overview of what's going on, we started with a diploid cell, right? So one diploid cell, that's going to divide and give us two identical, and if they're identical, right, they have to be also diploid like the original, right? 2 identical diploid cells. Okay, in meiosis, we're also going to start with 1 diploid cell. Okay, and so then we're going to end up with, actually, and you'll find this out in ANP2, 4 haploid cells, and they are obviously not going to be identical. All right, so they're going to be slightly different from each other. All right, you're going to end up with 4 haploid cells. Okay, so that's very different from mitosis. So mitosis, you are making exact copies of your original. Meiosis, you're not, right? You're making cells with half the amount of chromosomes because those are going to be your sperm and your egg. Okay, so that's the extent that I want you to worry about for meiosis, right? That's going to be for a different semester. So just be able to contrast it to what you learned about mitosis in this chapter. Okay? All right, so that wraps up this chapter. All right, and so you have your exam to study for, so make sure you're filling out your chapter reviews to study for your exam.